signaling the wound to progress through the next phase of wound healing. Our in vitro data even shows that they are capable of releasing both pro and anti-inflammatory cytokines. Lastly, the 3C patch does have published data on antimicrobial activity on Pseudomonas bacteria, which can potentially limit biofilm in wounds. So let's talk about the role of platelets in wound healing. We know based on our in vitro evidence that there's over 800 different proteins, cytokines, and growth factors that are being released from the 3C patch. We have platelet-derived growth factors, which are known to stimulate granulation tissue, transforming growth factors alpha and beta, which are increasing keratinocyte migration and collagen production, as well as VEGF, which is stimulating an angiogenic response and granulation tissue formation. We know that these growth factors are responsive and alive for multiple days, but the 3C patch is recommended to be reapplied weekly to get the max efficacy from these growth factors. So the 3C patch does have strong clinical evidence backing its efficacy. Today, I'm just going to highlight our largest RCT, which was published in the Lancet Diabetes and Endocrinology. This was an international multi-center randomized control trial that included 269 hard-to-heal diabetic foot ulcers. The exclusion criteria for this was very high, and it was focused in on hard-to-heal DFUs that had failed to progress by 50% in a 4-week time frame. So the 4-week run-in period was compared to the best standard of care on the market. This gave the 3C patch a 58% relative increase over the standard of care group, improving the chances of healing by 89%. I can share this study with us at the conclusion of today's presentation. So one area we know the 3C patch can be very beneficial is when treating ulcers that are probed to bone. This slide summarizes a series of 26 ulcers in 22 patients that were treated with the 3C patch. The wound ulcer duration was above 26 weeks prior to treatment, and the 3C patch treatments averaged around 8. We know that the 3C patch treatment resulted in bone being covered in over 70% of the wounds, and 58% of them had gone on to complete epithelialization. So in summary, the 3C patch, again, is 100% autologous. There's no additives or reagents, no contraindications, and it is indicated for the use on all exudating wounds. It does have increased healing for diabetics with non-healing wounds, even in probe-to-bone cases. I would say that the clinics of focus would include your wound care clinic, podiatry, vascular plastics, and even dermatology. And next steps for implementing this treatment at your sites would be to contact your local representative, which they can provide clinical evidence and centrifuge training. Additionally, I've listed the FSS contract information here for ordering. Great. So if time allows, I'd like to just walk you through a live demonstration of the 3C patch creation. Here you can see we're doing a venipuncture, collecting 18 milliliters of the patient's blood into the 3CP vial. It's as simple as just clicking it into the centrifuge, closing the lid, and pressing start. There will be a readout for how much time is left until your patch is complete, but the lid will open when it's finished, and at this point you have an entire hour to apply the 3C patch. Here you just take off the pink lid, and then you're going to apply the 3C patch waffle side to the wound. Waffle side is a side that has the impression from the filter. And you can see the handability of the patch is incredibly easy to work with. For demonstration purposes, here they're just scraping off the living cells and showing the tensile strength. Again, this is not a liquid or an injectable PRP product. There is some tensile strength. It's durable. It can be fenestrated if needed, but it's, again, very easy to handle. So I apologize, this video is playing a second time if you want to fast forward. There you go.
Okay, great. Thank you, Kelly. So I think we have some questions coming in, and if the people asking questions would just like to unmute, they can ask them in person, but I can read the first one, which was from, and I apologize if I mispronounce people's names, and Nicanor Moldovan. Kelly, for you, how does the 3C patch work in diabetic ulcers versus normoglycemic pressure ulcers?
Yes, so when launching a 3C patch, Reaplex actually really collaborated with CMS. This way, when they were doing their randomized control trial, they could set parameters to launch in the US market with reimbursement. So right now, all of our clinical evidence is focusing on diabetic foot ulcers. And we are seeing a tremendous response. Based on our randomized control trial, we know that patients can receive up to 20 applications once a week for 20 weeks. But the national average was under 8 applications to reach full closure on wounds that had been open for weeks to months. So our diabetics are classic, patients where this product really thrives.
So I thought I had an associated question, which maybe you answered and I missed, but how long does the 3C patch stay in place when you said multiple treatments?
Yes. So it actually, it'll stay in place and reincorporate into the wound. Our in vitro data shows that those growth factors are going to start to kind of die off around day 6. And that's why we do recommend weekly applications to constantly have the patch and growth factors present.
Okay. And Fabian, I think I have the same question for you. How long does Poiskin last? Because it looks more like a skin graft.
Yeah, our goal was to that Poiskin works as a skin graft. So we have not started yet clinical trials on human, but on mice we observe Poiskin after as long as we look at we follow up the observation after grafting.
Okay, and there was a question from Kimberly about vascularization of your heart constructs, but I also had a question about vascularization of the skin. Have you looked into doing that at all? Because that would really help with deeper wounds.
Yeah, so by design, we define the thickness of Poiskin so that we don't need to have a kind of prevascularization of Poiskin. So meaning that the thickness of Poiskin is not too thick to absorb some cell death, but not too thin to avoid contraction. So, but for deeper defects, as far as I understand, it's not so clear if it will be important to have a very thick Poiskin, for instance, or either it could be used in combination with some biomaterials or medical devices that are already used in combination with others. For instance, in combination with autograft. So definitely our goal just is to provide us an alternative to autograft.
Okay, and have you tried this with burn wounds at all?
Not yet. It's our plan in the clinical to include also burn people.
Okay, great. And I see, Jason, did you have a question, or did you just decide to turn your camera on?
Oh, no, sorry, I don't have a question. I'm just participating visually. Thank you.
Okay, it's the way my Teams screen works up. You're actually bigger than anybody else.
Wow.
I don't know why. I don't know why.
Got a promotion there. Thank you.
Yeah, I've no idea how Teams works. So,
hi, this is Shantae. Hello, everyone. I'm Shantae McCoy Jones. I am the VHA, Office of Nursing Services, Clinical Nurse Advisor for Pressure Injury Prevention and Management for the Department of Veteran Affairs. So I oversee all skin and wound for the VA and along with our ostomy as well. So I help to set the policies and practice and so forth. So I have a couple questions. So one of my questions to Fabian, so just to get a understanding is that you're going to now do the bioprinting at one of the hospitals. You're going to come out of the lab and now trial this on actual patients, correct?
Yes.
Yeah. Okay. And so when you had the mice, when you were practicing or in the lab with the mice, you were pretty much creating the wound from either how did you create the wound? Like, was it that you just cut the tissue and just pulled it back? Like, how did you create the wound for when you were doing your trial, your study?
That's a tough question because I have not performed the surgery by myself. Okay. So how it was, I don't remember exactly, to be honest.
And
sorry for that.
And the reason I'm asking that question, because I'm also a wound specialist, so I'm asking the question because I'm thinking about how this tissue, how this biofabrication tissue will respond based on the tissue injury itself. Like if it's related to a pressure injury, if it's related to a diabetic injury, if it's related to a burn, if it's a chronic, type of wound. And so that's why I'm asking that question, because I can't wait to find out what your results are, because based on how the tissue is injured, based on each one of those types of wounds, the body heals. I mean, the healing process is the same, but the level of damage, the degree of tissue loss and so forth is different. Burns do one thing where pressure also do something else. So in each one of them get to a level of healing just a little bit differently based on either what we put on the tissue or how the body's responding to other products and so forth. So that's why I'm asking that question, just as the thinking. And so that's my first question. The second question is, if you're looking at, say everything goes well, and you're looking at, okay, we're gonna start either renting this biofabrication machine or selling it. Has your company thought about a cost? Because I saw the slides that says a decrease in cost and affordability. Have you thought? Do you have an idea right now, or you're not willing to share how much that would be to a healthcare system or a hospital? Just curious.
Yeah, we have several. We have already sold a printer at Creatio, so meaning that this center has bought it, the clinical grade. We have sold many of research grade bioprinters that we are now installing, namely in Europe, probably in September in the US. But for the clinical grade bioprinter, today, what we propose to a cell therapy center, it's either that the center buy it, or we can propose also a kind of rental, because we know that, such kind of high-tech robotic system could be quite expensive. Sure. So we do think that moving towards a kind of by equipment as a service model that also has been implemented in other areas around medical equipment. We do think that it could be a good strategy, both for us but also for our customers, our partners.
Okay. I had sorry, Shante, I just had an associated question with that, though. Yeah. You mentioned that your machine is in GMP facilities. Most hospitals do not have GMP.
Yes, but there are numbers of hospitals that have now GMP facilities. We have, at least in Europe, that we know better than the US for sure. Sure. What we have in mind, and we have also this discussion with Professor Wheaton and so on, is that most of big hospitals that are in the center of a population of about 5 to 6 million inhabitants are now equipped or on the way to be equipped of a cell therapy facilities. At least, for instance, in France, that is not so big. It's about 60 million inhabitants. We have about 12 different cell therapy centers. So we know that other centers are now, or other hospitals are now on the way to establish such kind of center. But if you want to make this center sustainable, it's important that they are able to produce different kind of cell therapy products or therapies. So it's important, at least what we have in mind is to enable them not only to produce skin, but also other products.
Okay.
But probably Burns and Wound Care Center will be very focused on skin, but other centers will be more will produce
more general.
More general,
Yeah, Nicanor,
And
I see you have your hand up.
Well, I had another question for Kelly because I was
Okay.
Yeah. So Kelly, nice to see you. Glad you could join us. Question for the patch which you're sharing with us. So replacing it every 6 days, well, once a week. Is there a time period where if you don't see a result that you recommend that another method of wound healing is used? How do you set the parameters with that facility once you go in, you do the education training? How do you then ensure that the application of the product is consistent with what the procedures and standards you set forth for it? So that's one of, because that's sometimes people get caught up with, okay, I'm gonna keep using it, and after 5 weeks, they're not really seeing anything, but they're keep using it. So how is that for this product?
Yes, it's great to see you too. I'll see you in Philly one day soon. But that's a great question because every wound is going to vary, and 3C patch is indicated for any exudating wound. Earlier, I was just stating that our clinical evidence really focuses on diabetic non-healing wounds. But in general, our sales force is trained to help sites understand that if you're not seeing a response by the 3rd application, then maybe we step back and reevaluate. It does work rather quickly. The first week, the first application, and even second application, there's a lot of building behind the scenes, and you all know that in wound healing. But by the 3rd application is really when things start to really organize and show some kind of response to the clinicians. So I would say by week 3 is our prep.
All right, thanks.
Great, thank you. Okay, Nicanor, you still have a question?
Yes, I thought it's good to invite Niloufar to present herself because it's the connection between Poetics and Northern America, right? We had the pleasure to hear her last year, so welcome again.
Yes, hi everyone. My name is Niloufar. I'm the product manager for Poeti's next generation bioprinting system in North America. So yes, last year, for those of you who attended the symposium, I presented on Poeti's behalf. So, but this year we managed to get Fabien, hear everything from him firsthand.
That's great. Thank you.
Well, yeah, I hope we stay in touch, and we would be happy to contribute to all these promising projects moving forward. So please stay in touch with us, or if you have any questions, if inquiries, please let us know.
Sounds great. So do we have any other questions from the audience?
If I may add one more or two more. For
Fabien, of course you may.
Regarding the integration of the printed skin with the existing skin, do they eventually fuse in any way, or they will continue to coexist separately?
Would you repeat your question, because you mean what would coexist, skin on polyeskin? What would you?
So if the printed skin integrates with the recipient's skin, and if it is a true replacement of the missing part with a functional new one, considering there are no blood vessels, I was wondering if it is possible to see such an intimate integration, or it continue to exist like a scar on the surface of the wound.
At least what we have seen on studies on mice is that we observe some vessel formation through polyeskin. So it's probably based on, I would say, on design, because we have not performed yet as a clinical trial. We expect that polyeskin will work as a splint skin graft, which and for what we know is that for not too sick samples, we observe a vascularization after grafting. So we expect the same mechanism of action, I would say.
Okay. Dante seems to have another contribution.
Yes, I'm gonna add to your question, which you leading me right to. And what I am not a lab scientist, so I don't know how my skin looks. Okay. All I know is when I run. So my question is, in answering that question, have you seen any changes in pigmentation? Especially, I don't know how the population is where you're going to use the your biofabrication at the medical center, the hospital that you're going to use it at, so forth. And looking at melanin in the skin, the repigmentation for a population. So if you're dealing with someone who has darker pigmentation, you put this patch on them. You know, have you seen any repigmentation? Are you seeing any, not melanin, but I don't know what you call the cells for mice. Have you seen any color that becomes similar? I know you're saying that it does revascularize, but I'm also thinking about the color repigmentation as well.
Yeah, we have not studied that yet on since it has not been implanted yet into human. And it is quite difficult to look at least to my knowledge, to look at it into mice. But what we have under development is to also integrate some melanocytes. But it's also a way to be able to isolate or to have the right population of melanocytes. Because It has been quite well known now that the protocols to print melanocytes into full skin construct is already known. But probably the main challenge is to get the right population.
Okay. Thank you.
There was an associated question about hair regrowing and sweat. Does that happen in the patch, or is it not?
It would be probably difficult to expect some hair growth, except if we integrate some specific stem cells and so on that are known to promote the formation of hair follicle. In the past, we used to work in vitro on hair tissue model, namely with L'Oreal, but the protocol are not really the same if we look at if we deal with clinical applications.
Okay, but would the patch remain? It wouldn't have any sweat glands. So
no. Well, it's also a matter. I remember that Professor Bonassar mentioned that the goal for him was his strategy was a low-hanging fruit strategy. I would say we use the same, meaning it was impossible to, at least to me, on a company like Poietis, to have as a first product a skin tissue with sweat glands, hair follicles, vascularization. And so
on. You've got to start somewhere.
It's a too high complexity, even in terms of cell biology. So our goal is to go step by step. And the first step was to establish an autologous skin substitute with a dermis compartment on without vascularization, without air follicle and so on. But
Okay.
it's already a quite big challenge, I would say.
It certainly is, and it's very interesting to see how both of you are meeting these challenges. So I think we're pretty much out of time for the current session, and we'll move along to the next. I believe that's being moderated by I had that up in a minute. Now I'll pass it over to Sandeep. Is that correct?
Yes.
Yes.
Thank you all the speakers of this previous session. And the next one will be dedicated to organ biofabrication. The moderator is Dr. Sandeep Malipattu, who is Professor of Medicine and Chief of the Division of Nephrology and Hypertension of Renaissance School of Medicine at Stony Brook, and staff physician at Northport Veterans Affairs Medical Center. The focus of his research is on molecular mechanisms mediating the development and progression of kidney disease. The laboratory concentrates on the critical role of a family of zinc fingers transcription factors and on developing a combinatorial approach to build a platform for kidney regeneration by using high throughput screening strategies to identify small molecular compounds as novel therapeutic agents. In one of his recent papers, he also reviewed the current status of kidney bioengineering, including 3D bioprinting, thus indicating an interest in this new direction. So, Sandeep, please take over leading this session.
Well, thank you for that introduction, and it's certainly a pleasure to moderate this really exciting session. I think we have a panel of really exciting speakers, and I think just as a reminder for everyone, please post your questions in the chat as we go through it, or save them certainly till the end of the Q&A session. And we can kind of get started. And I apologize if I'm going to keep the introductions brief for all our speakers that are highly accomplished, but I apologize for that, just so that in the interest of time, they have an opportunity to present their work, and then the question and answer session at the end. Our first speaker today is Dr. Adam Feinberg, who's a professor of biomedical engineering and material science engineering at Carnegie Mellon University. The title of his talk is on fresh 3D bioprinting to rebuild the human heart. Dr. Feinberg is the principal investigator of the Regenerative Biomaterials and Therapeutics Group, really founded at Carnegie Mellon University in 2010. His laboratory focuses on cell material interactions and understanding how guidance cues can be encoded in extracellular space to organize multicellular assembly. Here, certainly the focus of the talk is going to be on the heart, but it certainly works on other systems. So I think we can start the presentation.
Can I confirm, can you see the presentation at this point?
Yes, we can.
Okay, perfect. And you can hear me okay?
Yes, perfect.
Excellent. Great. Well, thank you very much for the introduction, Sandeep, and also just thank you to the organizers for the opportunity to share some of our work today. I'm a professor at Carnegie Mellon University. As was mentioned, I'm also, I want to disclose that I'm also a CTO and co-founder at Fluidform Inc. It's a startup company that's focused on commercializing the fresh bioprinting technology that I'll tell you a little bit about now. This talk is really focused on how we're trying to leverage more complexity to build larger scale, more functional tissue constructs, and ultimately one day organs. Really some of the steps we're taking to get there. But the heart is really just an example. You can think about kind of a similar story for many other tissues and organs in the human body. So, one of our goals is to ultimately get to therapeutic scale tissues and organs, really towards, repairing solid organs, the failure of which ultimately leads to death in a large percentage of the population. There's a number of things we've been trying to get to along that path when we are developing new biofabrication capabilities. One of them has really been trying to get large volumetric scaffolds where we can recreate anatomical structure. Having mechanical strength and robustness, I think we saw that some great examples of that in the previous talks around the use of wound skin regenerative products where you need a certain durability. Heart valve is another good example of that. You know, if we're building volumetric tissue, we have to have vascularization. The talk by Dr. Bonaster was great about cartilage, where vascularization is really not wanted, but most other tissues, that's really a critical foundation from the micro to macro scale. And then ultimately, again, for many solid organs, we want very high cell densities on the order of, you know, 200 to 500 million cells per ml. That's at least 1 to 2 orders of magnitude higher than most bioprinting has been conducted to date, although not in all cases. So one of the challenges we've really worked to address is just making 3D bioprinting even work with soft hydrogels and cell-based materials, especially when we want to go big. So your standard 3D printer shown here is a layer by layer technique that we all know that works really well for most metals and ceramics and polymers, because when you put a layer down, it's relatively mechanically stable and it makes it easy to put the next layer on top. And then repeat as you go up. That's a lot more difficult for hydrogels, right? Because they're soft, they deform under their own weight, they have material properties similar to Jello. And as you build up layers and layers under gravity, they start to deform. And then once a layer deforms, then you can't accurately put the next layer on top as you go in 3D. And if you're also dealing with cells that are actively moving, that's a whole nother challenge of keeping a layer stable during the fabrication process. So we developed a technique that we call FRESH. FRESH stands for Freeform Reversible Embedding of Suspended Hydrogels. And if you look at this cartoon, the real secret here is that we simply print a hydrogel inside of another hydrogel. And that's how we basically are supporting these soft materials during the fabrication process. So we have a gelling fluid bioink that we extrude into a sacrificial support material. That would be the yellow material in this example. That support bath behaves with a yield stress fluid, which just means it transitions from solid to liquid with applied force, which is why a needle moves through easily, but whatever we extrude stays embedded in place. That bioink is uniformly supported during the printing process, and when we're done, we simply raise everything from room temperature, where we do the fabrication, to body temperature. That releases and melts the support bath and relieves whatever we printed behind. So our support bath has this yield stress behavior. It's very much like hair gel or ketchup or mayonnaise, right? These are materials that flow under load or are otherwise solid. And here's just a video of this process. Hopefully this comes through on Teams, but you're seeing us extrude alginate, which is the darker material. Alginate will cross-link due to calcium ions in the support bath as soon as it's extruded. And then we're simply raising the temperature when we're done, and melts the support bath, releases whatever we bioprinted non-destructively, really tuned to be within a range that cells and proteins are quite happy. And this is an embedded printing approach, and I just want to highlight, we're one of a number of labs that have really pioneered this approach. Jennifer Lewis and Jason Burdick and Tommy Angelini are others. I just don't have time today to kind of cover the broad spectrum of others in this space. But we've been really exciting that FRESH has really grown as a biofabrication platform. You can use a really wide range of bioinks. That's because the support bath doesn't just support the bioink, but because we print inside of an aqueous environment, we can use the environment to do biochemistry, which means we can use a wide range of cross-linking mechanisms, and then we can also change the support bath. Really, the take home here is that we are almost unlimited in the geometry and types of bioinks or biomaterials that we can 3D print in the system, far greater than any other biofabrication approach that we're aware of. We're also not limited to layer by layer. We can actually freeform dispense material in space, which means we can start to recreate some of the complex anisotropy we see in most tissues. Because, while skin is mostly a layer kind of structure, most other organs in the body are not simple layer by layer tissues, and therefore we need more complexity when we think about recreating the structure and function of those. So I just want to give you a few examples. Some of these were published in our Science article back in 2019, and a few more recent examples. This is looking at building functional cardiac muscle tissue. So here we're printing collagen with one bioink and cells, which are human cardiomyocytes, in a second bioink, into a very simple elliptical cup to model a left ventricle. We can actually align the cells in the direction we extrude them during the 3D printing process. And if we look at one of these beating ventricles under the microscope, you're seeing the fluorescent signal, which is the calcium during each heartbeat. You can kind of saw it moving in a wave. Here we're field stimulating it so that all the cells synchronize together. That's a little boring when we watch it from the side, but in one moment we're going to change views and look into the chamber. You can first see a spontaneous wave, but when we stimulate the contraction and synchronize the cells to beat together, we actually get wall thickening and decrease in chamber volume. So far less than the native heart, but the tissues are at least starting to function in the same way as the native tissue. We've been able to also show that we can use the same technology to print collagen with really incredible mechanical properties that start to rival the collagen properties of native tissue. So here we have a heart valve. It's a trileaflet heart valve, basically like an aortic valve. It's entirely printed out of collagen. In this case, it looks like it's rubber, but it's actually 100% native collagen. Here we have it in a flow system that models physiologic flow rate and pressure, showing that we can maintain and support physiologic flow. And then we can also scale this technology beyond the valve to the whole heart. So here we're going to see a micro CT image of a 3D bioprinted heart scaffold out of collagen. This is an adult human heart based on an MRI image. We actually scaled it down to the size of a newborn neonatal heart. But what you can kind of see is the fidelity. And these holes in the heart are actually not really holes. They're just due to lack of X-ray contrast for how thin the collagen is in certain areas. But as we kind of move through layer by layer, we can see the different chambers of the atria. We can move down and see the aortic and pulmonary valves. And as we go lower, we can see the ventricles. So what we can actually do is now start to create, large 3D constructs. We've been really excited to see Fresh grow as a biofabrication platform, even just in the area of the heart. Great work by Tal Devere of the University of Tel Aviv, creating a cellularized heart using human IPS. Brenda Ogle of the University of Minnesota demonstrated a beating chambered organoid where they're actually able to generate pressure volume loops. Jennifer Lewis developed a modification of this approach called SWIFT, where the support bath itself was made up of cardiac spheroids that they could print inside of and create structure. And then we've actually been able to show that we can scale this all the way up to the whole organ size. So this is a full-size human heart printed out of alginate. Mostly because collagen is too expensive at this point. It would have been a $15,000 heart. And this is still something we can't cellularize because we can't generate the 10 billion cells we would need to do that, but that's where we're going. What we are doing today is building human hearts, but at a simpler scale. So really taking a cue from developmental biology, where early on the heart is actually a tube. So we've been trying to build that earlier stage of the heart. Here you actually see one of these heart tubes. It's beating here under stimulation. And here's actually one of these heart tubes actually contracting. This is about 1 centimeter long and 1.5 millimeters in internal diameter. We already have it pumping beads shown here, where we're tracking beads to show the flow. This is without valves, so the beads just kind of go back and forth. And we can quantify that. But some of the work we're doing now is trying to move these into rodent models as our initial work in building functional hearts for in vivo implantation. So hopefully I can talk about that at a future discussion. But we are also taking an eye towards clinical translation. But here we're realizing that building a cellularized full-scale muscle tissue, whether it's skeletal or cardiac, is beyond current scope. And so what we're trying to do is create structured ECM scaffolds for volumetric muscle repair. And what you simply see here is basically a CT of a patient that had a blast injury to one leg. But what we're able to do, similar to others, is take the contralateral leg and create a muscle construct that recreates the missing tissue void. We can then fabricate that entirely out of decellularized extracellular matrix-based bioink that we can then implant for regeneration. So these are not yet in humans. These are still in preclinical large animal studies. But what I want to point out is these are about 14 centimeter long scaffolds in the longest dimension. So really, I think the largest 3D hydrogel scaffolds anyone is currently trying to biofabricate. And again, Fresh easily allows us to go this big. Just in the closing minutes, I do want to highlight, we do something really unique in my lab, in that all our bioprinters are developed on open source platforms. So we run a course at Carnegie Mellon University where you can come to Carnegie Mellon, build your own bioprinter and take it back to your home institution with you. We do that every year. We also, if you look down here in the highlighted yellow, we actually took that course and turned it into a manuscript. So you can actually download this and do the whole course at home or at your home institution, and also build your own bioprinter that outperforms most any commercial one for less than $1,000. And we've had a large number of collaborators really across the globe that have participated in this over the years. So just to try to just to summarize, we're really trying to advance these tissue engineering applications with more complex models. I haven't shared the vascularization, but we've made a lot of advances in vascularization from micro to macro scale. We've been integrating biomanufacturing platforms and advanced 3D imaging. We're translating some of this to large animal preclinical models, so that human example I showed is already doing that. We're supporting the research community. One example is through those open source bioprinters. And then, as I mentioned, that when we started, I have a startup company where we're developing commercial applications. So I just want to really quickly thank the people who did this work. I have an amazing team of postdocs and grad students and undergrads. I obviously don't do anything. I just get to talk about the amazing things they do. They're the ones every day making it happen. And then also my funding sources, particularly want to highlight Additional Ventures Foundation for the cardiac work, and DARPA that's helped fund some of that volumetric muscle work. So thank you very much. And we are hiring both grad students and postdocs, so feel free to apply if you're interested. So thank you.
Thank you. That was excellent, Dr. Feinberg. So I think we're going to hold the question and answers to the very end. So if you could stay on. So moving to our second speaker, I'd like to introduce, we have two concurrent speakers, Dr. Jason Wertheim and as well as Dr. Kyle Rockhaus. Dr. Wertheim is professor of surgery and biomedical engineering and vice dean for research and graduate studies at the University of Arizona, Southern Arizona VA healthcare system. Dr. Kyle Koss, postdoctoral fellow that's worked with Dr. Jason Wertheim in this area, and the title of the talk is on kidney and liver bioprinting. The laboratory focuses on really developing biomaterials for tissue engineering applications, as well as investigate the growth and differentiation of cells into mature tissue, liver, kidney, and vascular tissues as it relates to within the biomaterial scaffolds to model disease or develop cells for therapy and tissue transplantation. I guess we can start the talk.
Great. So thanks so much. Let me just introduce, Kyle. So Kyle's a senior postdoc in the lab, really doing great work. So I'm going to have him give the bulk of the talk today. I'll be around to answer some questions at the end. But the work kind of really centers around some of the new technologies that are kind of on the cusp in our group in 3D printing and scaffold development. So I'll let Kyle take it away. Thanks a lot.
Thank you. Thank you very much.
Yeah. So do I have my screen shared? Can everyone see this? Okay, good. So hi, everybody. Like Jason said, we're going to talk a bit about some of the early developed technologies, and sort of my expertise lies in the manufacturing of a lot of biomolecular tools that we're working on in the lab, and some of the novel printing techniques that we're working on. So some of the work we're previously doing, and then some of the work we're kind of proposing what we'll do with some of these printers we have. So I think most folks are familiar with iPSC technologies nowadays, and in our work we're really focused on trying to print the liver and the kidney, and in this talk I'll focus mostly on our kidney work. And so in this figure here, you can see that there's a lot of stages of growth, and we work on organoid systems to get these sort of final stages of specialized subpopulations of the kidney. And in this case, we're looking at like the glomerular system, the distal and proximal tubules. And
Kyle, you may want to check out I'm seeing something different. I'm not seeing your slides there, so.
Yeah.
Yeah, I can play your recording. Do you want me to play your recorder, or are you going to be live?
You had them before and they flipped off, so
yeah.
You may just have your other screen shown.
Okay, I can share that.
Okay. Is just going to run my recording?
Yes.
Okay, we can do that. Yep, that's fine.
You prefer to go live? Okay.
Tools are. My name's Kyle Koss, and I work with Jason Wertheim. And in our lab, we attempt to generate artificial organs such as kidney or livers. I'd like to give a talk on what our lab's tools are and the techniques we're developing in the generation of, in particular, kidneys in this situation due to our time limitations. So most folks are familiar with stem cell differentiation and how you can take a stem cell from a person, generate an iPSC, and go all the way to generating certain structures or certain cell types. In this case, in the kidney, we're really interested in the cells that comprise the glomerular system or distal proximal tubules. But we're also interested in taking the microenvironment and looking at that and how, these could recapitulate 3D inks or D-cell scaffolds, and what are the characteristics that we need to get the cells in the right areas of those prints, but also behaving properly in the correct phenotypes. So our primary work was based upon D-celled scaffolds and recellularization. And so this is typically an organ from an animal, in this case a rat, for example. And you can you can add detergents and surfactants to strip the cells out. And you can see the vascular ECM is still present. And then you can reperfuse with blood to recreate the same type of flow with minor leakages. And you can actually reperfuse to recreate that vasculature with endothelial cells or HUVECs. Now, there are some problems with this, such as thrombosis, due to the fact that these don't really form the tight junctions typically seen in tissues. And so we're working on ways to deal with that. And so this is an example of how one would take a kidney and remove all those cells and use tritons and SDS surfactants with peristaltic pumps, and you can remove all those, and then you can replace cells with the same type of pumps and put them in, house them in an incubator in a reactor system like this to actually generate the artificial organ. And the cells that you'd be perfused would be taken from components of your organoid, and this is an organoid that we generate in the lab. And as you can see, it forms, it differentiates as a cluster of cells, and each of those have populations of pockets of interest. In this case, you have the green, which is your podocytes, your red, which is your distal, and your yellow, which is your proximal tubules. And so one of our main projects that we're working on is, of course, every organ requires vasculature. And so what we're printing is we're trying to recreate that vasculature. And so we have a simple print in this situation that we're trying to use our 3D bioplotter or our VisionTech printer with the idea of a spiral print and an inner layer that we generate with a collagen or some type of self-assembling surface, and that we can actually use some sort of targeting molecule to localize in that inner layer and then generate an endothelium very similar to tissues. Now, there are some challenges that we're working on with that, and the major concept is, can we create a system that has the flow, which we've seen with D-celled scaffolds, but can we also create something that is filtering? And the idea is that we have this complicated system with podocytes and endothelium and certain basement membranes and a lot of factors that are key to that area. And so it's not necessarily certain whether if we print these cells side by side, we're actually going to get that behavior. And so the idea is we want to do something like enrich the area with certain growth factors or certain molecules to either prevent that thrombosis I mentioned before, or to actually enhance the cell behavior a certain way. And so one of our major strategies is we have these ECMs present in the inks or coatings or in those other areas, and we have a targeting region like a collagen 4, and you can see there's a peptide here that we're using that's specific to collagen 4, but we also have a bioactive region, which could be a growth factor, some other type of bioactive molecule. And so one of our first major projects was looking at heparin. So heparin is anti-clotting, and we added this with NHS-CDC chemistry to add the collagen binding peptide. And in this situation, we're looking at a reduction in thrombosis and this type of scar tissue formation, neointimal hyperplasia. And so we were successful at putting that into an animal model, not only seeing proper localization, but reduction of that scar tissue. And this would translate over to our printing technology as well. But what we're also doing is we're finding new binding motifs of our own. So rather than using what's pre-existing, we're looking at peptides that might bind to hyaluronic acid, which is also a high component of a lot of 3D printing inks like HAMA. And so what we've done is we looked at these helicodomains in RAM, which is one of the major receptors for binding to hyaluronic acid. And these domains are called BX17B domains that are present on the outside face of the helix. And we plotted that in a 2D manner, which we used to optimize multiple binding domains and recreate several peptides of our own, which are quite high performers. And hopefully we can use these in some of our technologies. Furthermore, we're looking at modifying our binders with growth factors such as VEGF. and also peptides like QK, which are analogs of that. And we're also looking at using the biotin-avid interactions to mix and match different growth factors to different binding motifs. So we can add the hyaluronic acid versus the collagen binding peptides, or maybe an angiopoietin versus a VEGF. And what's more, we're actually looking at peptides that could be tetramers with multiple biotin sites, and avidin is a tetramer itself, which means you have a co-assembling event. So if there are issues in binding, we can actually go beyond the 2D limitations of binding sites on these ECMs and grow our coatings to a more significant way. And you can see we have some results of that with our heparins. So one of the other issues that we have is we want to really optimize our prints to see if these interactions work in more sophisticated ways. And we're fortunate enough to have these specialized printers, or nanolithographic printers from TerraPrint. And we have these tandem printers that are very complementary. The M series, which is a contact print. You can see on the bottom left cartoon there that this is just an array that makes contact, and there's a chemistry when that happens. And in the TerraPrint E version, you have a beam pen lithography, which you have tiny apertures in your array, so that makes a photochemistry, so you can actually utilize this with a lot of inks that are comparable in your 3D prints, like your methacrylation chemistries. And you can see that you can make fluidic chambers, and you can pattern very specific cell binding sites, or mix and match multiple different types of chemistries. And you can see that these are nanoscale in nature, so you have your probes that are 60 nanometers and your apertures, which are 250 nanometers, and you can generate nanostructures rather than microstructures, and or microstructures as well with both, and you can really optimize your replicating biological systems. And so the M series, which is our contact chemistry, primarily functions based on ethylation or a sulfhydryl group, and when that makes contact, it covalently links whichever group happens to be on the other side. And in this case, typically you would use a MHA, which has a carboxyl site. You could use various chemistries like NHSCDC to build proteins and peptides onto that. But what's more, you can also etch the gold in areas that weren't printed and actually depth-wise etch into the patterns to make nanoporous systems or pseudo 3D structures so that your cells can interact with. And in the case of our beam pen lithography, which is our photochemistry, it utilizes what's called a micromirror array, and you can turn off any one of these mirror systems to pattern something very precise. And in this case, we're using it to potentially look at individual layers and the methacrylation. So in this case, we have a gelma. We're patterning an 20 micron individual layer of cells. And this allows us to potentially look at cell-to-cell interface with specific ways of localizing different types of proteins in different areas, which could recreate a more proper GBM, or glomerular basement membrane. But what's more, we can also utilize these machines to nano-pattern different types of molds for a fluidic system. So we can actually utilize this to recapitulate our flow systems, in our case, our GBM or nephron. So we're looking at a machine that's quite diverse in this case. So as a summary, our lab really focuses on developing the organoid system. So we want to recreate the right cell types. We're also looking at generating biomolecules and conjugates that we can use to engineer interfaces in our 3D prints, which we're trying to recapitulate with our decellularized systems. But also, we're using a nanoprinting system to really optimize and develop our prints and patterns so we can go back to the 3D printer and know that our interfaces are proper and they will function well. And with that, I'd like to thank all the wonderful folks in our lab and our excellent funders and associates. And with that, I'd like to open the floor to questions and discussion. Thank you very much.
All right. Thank you for the excellent presentation. I don't know, Dr. Wertheim, did you want to add anything additional as well? Or wait for the question and answer session? You're on mute. I think you're on mute, Jason.
Yeah, sorry, I
can't
had a
hear
little
you.
bit of internet issues, but I think Kyle summed up very nicely the cellular and bioprinting work. So thank you.
All right. Again, just as a reminder, please post your questions in the chat and also at the end of the session. So we'll move on to our third and final speaker, Dr. Angela Panoscolatis Mortati, and she's PhD and professor in pediatrics in the Division of Blood and Marrow Transplant and Cellular Therapy, as well as pulmonary medicine at the University of Minnesota. She's also director of Cytokine Reference Laboratory, as well as the director of 3D Bioprinting Facility at University of Minnesota, and vice chair for research for pediatrics. Her research really focuses on use of stem cells and tissue organ engineering and biofabrication, and especially in the context of decellularized whole organ scaffolds as bioreactors, as well as using 3D printing, bioprinting, using extrusion, suspension, and laser-assisted techniques. Title of her talk today is on lung and hollow organ bioprinting.
Thank you so much. Can you see my presentation view?
Yes.
Okay, great. And I'd like to thank the organizers for inviting me and for the opportunity to cover this massive topic in 15 minutes. Have some disclosures. Some portions of this work are subject of published provisional patent applications, and then some other that are recently, and by recently filed, I mean like just a few days ago and even last week, some. And so I'm going to be presenting things that are not published for the second half of this talk, because I felt that, speaking to an audience that's already well versed in this field, was trying to struggle with, what to present that would actually really interest you too. What I want to also acknowledge is that 3D bioprinting, of course, and other types of fabrication technologies are used in creating in vitro models, using spheroids or organoids and organs on a chip and nanostructured biomaterials. But I'm not going to be talking about that because my charge was to talk about actual printing full organs. So first, lungs. Can we make lungs? And usually this is the structure that, people show when they're showing this, branched, highly branched structure. Usually it's got, 23 to 25 branched branchings all the way down to the alveolar area. So in white is the airway tree, and then on the left side is the airway tree, and the right side in color, the blue and red, is the vasculature. But we also, and if it was this, let me say simple in quotations, that would be pretty great. But unfortunately, there's a lot more to the lung, being the mesenchyme, the pleura, there's so much interstitium that it's really more like a sponge, and it's not so much just this bunch of, tubes going through the lung. And so we need to keep that in mind. And also, the fact that there are so many cell types that, people have heard about many times. But, we have not just, this whole range of different types of epithelial cells and progenitor cells that can help the lung renew, because it undergoes a lot of environmental insults all the time. But also, the innervation that is there, all the inflammation and the inflammatory mediators and cells. And again, the stroma and all the interstitium, but also how this hierarchy of cells changes as you go down the airway tree into the alveolus, which is actually the business end. So that's where I'm getting to this here. This is where air exchange actually takes place. And this is usually how the vasculature, the capillary network, where the exchange takes place in the alveolus is depicted. But what I've been told by people who've studied this a lot is that it looks more like a parking garage. This is how the vasculature really envelops that alveolus. It's more like this, where you have, these areas that are sort of joined, but then imagine blood flowing through this parking garage. That's actually more like how it's structured. And in fact, the lung has the largest surface area of any other vascular bed. So it's really quite, quite a challenge to create something like this. And the surface area of the airway tree, has been quoted many times. People say that it's the surface area of a large tennis court. So that's quite a remarkable thing. So the two main ways that people have attempted to create whole lungs is through the typical decell recell technique that you just heard about, and also using 3D bioprinting. And so my lab's done quite a bit of work on decellularization. And in fact, once you decellularize a full set of pig lungs here, and we've done this with human lungs as well, and ventilate it, you can see like there's no escape of air, there's no bubbling. It's really quite remarkable that the entire matrix stays intact. But how to create something like this? So we automated this whole system, as you see here. This is all work that's been published. This is just some more stuff, but it gets to be quite intricate in creating not just the decellularization chamber, but it also has to act as a recellularization chamber, and then more like a bioreactor too. So it really is quite a bit of understanding of physiology that's required here. And of course, the idea is, that, as you just heard in some other talks, is to keep the organ intact, keep this matrix intact, so at least you have that part of the structure already pre-created, and then use the patient's own stem cell derived cells to repopulate it and create an organ that way. And we've done some work on that, and we've had the most success, as have other people, in recellularizing the vascular tree. And when we were trying to figure out, okay, what is the actual endothelial cell type that should be used for this, we actually, in collaboration with Diego Alvarez, when he was at Mobile, Alabama, we got different types of endothelial, rat pulmonary endothelial cells, and that they had isolated from either the pulmonary artery or the microvasculature, and also a progenitor cell population they identified in from the microvasculature. That they called rat microvascular endothelial progenitor cell. It's this full long name here. And what we saw was when we infused into decellularized mouse scaffolds these rat endothelial cells, that the cells had tropism for the area from which they were derived, which was pretty amazing to us. So for example, if you took from the larger vessels, they had propensity to only repopulate the larger vessels, whereas the microvascular cells would repopulate the microvascular cells and not the larger cells. Whereas when we took this progenitor cell type, and you can see here too, it would repopulate the entire vascular tree. So that was really quite remarkable, and we determined the surface phenotype of these cells. So it includes typical markers of endothelial cells, but also some scavenger receptors and integrins, and also we looked at an array of binding to lectins to help further identify what this cell type is. So we think that in trying to restructure whatever strategy is going to be best for creating a whole organ. And it doesn't mean it's just for the lungs, but other organs too. We have to keep in mind what is the right cell population to use in order to, recreate the proper structure. And then we also are interested in ways of trying to non-destructively evaluate how constructs are behaving, how tissue engineered constructs are behaving. So what we did here was, in order to study the barrier function of these re-endothelialized lungs, we looked at thrombomodulin activity just by perfusing through the vasculature some alpha thrombin and protein C to measure thrombomodulin activity, and also looked at endothelial leak by putting in Evans blue dye and showing that the recellularized ones actually were closer to the fresh non-manipulated lungs as opposed to the D cell. So that was really encouraging for us in using these progenitor cells. So they behaved, the results for these were better than by mixing the microvascular cells, for example, with the pulmonary artery endothelial cells. Even mixing those two together didn't give you results as good as putting in just that progenitor cell population. So this, of course, then leads into, okay, what kind of bioreactor are we going to use? And there's been an array of them, and you can, read in this review paper that I wrote about all the different types of bioreactors that have been established by various groups. And, some of the top groups in this area, of course, were Laura Nicholson's lab, Harold Ott's lab, and a few others who have really done a lot of work on this. And of course, then that leads into the ex vivo lung perfusion systems. So this is the group from Toronto that has developed this system here, and then the one, of course, by Transmedics, which we had shown in some other work that you could recondition lungs that were previously rejected for transplant use, but they could be reconditioned in a machine such as this. But I'll also point out to everybody that, even when you have a perfectly good intact lung, you can only keep them going alive like this for like 24 hours max, maybe 48 if you're like really vigorous with your pressors and other, supplements in the system. So, that begs the question, okay, then, even if we're to engineer a lung, how are we going to keep it alive? So there's a lot that we still don't know about lungs. So let's keep that in mind. Now, as far as using 3D bioprinting to create, now I want to focus on inflatable perfusible lung tissue. Okay, I don't want to just say something's vascularized, because a lot of times people call vascularized just throwing endothelial cells in. Okay, that doesn't mean it's vascularized. Vascularized means, yeah, it's perfusible and contains endothelial cells. That's what it means to me. So this, of course, is Jordan Miller's pretty famous image that they published several years ago now in Science, showing how they use the FRESH method in order to create this construct that you could ventilate, put air in and out, and also create a vascular type of system around it, wrapped around it. But of course, it didn't reach the resolution required of that at the alveolus level. But still, it was very, very promising work, and certainly very exciting that this type of complexity could be achieved in that way. But people who've really, really, made the biggest advances in using 3D printing to really create a lung is the group at United Therapeutics using digital light projection-based polymerization of hydrogels. And so this is what their types of constructs look like. So it's the organ manufacturing group there, and they do this in collaboration with 3D Systems, which was, of course, co-founded by Chuck Hull, who's like the father of 3D printing in general back in the 80s. So these are some websites I encourage you to go to, but they were showing that using this type of method with human collagen, they could resulting in 50 trillion voxels being used. They could achieve subcellular resolution about 1 micron. It takes 3 weeks to print a whole set of lungs. No cells in it yet, just the whole scaffold. They were able to make 4,000 kilometers worth of pulmonary capillaries, as well as lymphatics. The lymphatic, structure embedded within this, 500 million alveoli, and they've been able to demonstrate gas exchange in animal models. Now, this is just stuff that they have publicly disclosed. So for sure, they've done a lot more. And so this is, the group that I think is just leagues ahead of everybody else in this area. So now I want to move on to hollow organs. And you've heard already some of these types of concepts. And, there's a review paper that my group wrote that encompasses sort of, in this figure here, a summary of a lot of other investigators' work about different strategies for creating hollow, tubes, hollow organs, such as vessels, trachea, esophagus, those types of organs. So, using different types of polymers mixed in with bioinks, and using, different types of architectures, using the Kenzen technique, of course, spheroids in the needle array, sacrificial molds, using on mandrels, either using decellularized ECM and rolling constructs, or electrospring, overlapping electrospinning and electrospraying on top of these types of constructs as well. So many, many different types of techniques, and I'm not going to go into them all in detail. I will say, though, that there are some other improvements that are required. And so my group, for example, in this paper, we showed that if we took decellularized matrix from the trachea and cryogenically ground it up into small microparticles so that they were like less than 50 microns in diameter, and incorporated that within a GELMA hydrogel, we could really increase the fidelity of the print. So shown here is a larynx, And then shown here is a bifurcating bronchus. So this is what, the result is of adding microparticles to this Gelma hydrogel, as opposed to just Gelma alone. And interestingly, you could also, so you could increase fidelity, but also you didn't need to cool the Gelma. You could do this at room temperature, which is, to me, a big advance, because that decreases the complexity of the manufacturing process. So then here's another example of, what many people have done, and that is to, print on a mandrel. So you can print, different orientations. So we've printed some here, around the mandrel, and now we're printing a longitudinal direction. And the flashes that you see are the flashes of blue light in order to cure the gel that's been methacrylated. And so then there's a photo initiator there to cure it. Okay. And then we're getting to another hollow organ, the gastroesophageal junction. And for this, we want to use pre-aligned microtissues. And here, our indication is that, most esophageal cancers happen at this junction. And the surgical solution is, of course, to cut it out and then do a gastric pull-up and then reconnect up here, closer to, your throat, which is not a good outcome. And there's, a really complex alignment in this area. And so we were trying to figure out how to get this, how to tackle this problem of this alignment geometry complexity. So we were thinking to create pre-aligned microtissues and then use those in the bioprinting process. And we do that through a spinning technique here. So here's a spinneret. So we have cells in the center and alginate outside, and then a calcium chloride to cure that alginate. And we can create these microtissues like this. And here it's just showing how the spinning process looks. And you can spin, we've spun things like 100 meters or 100 yards long, 100 meters long. But, people, if you have enough material and enough cells, you can spin like kilometers long of fibers. It's really quite incredible. Here's what it looks like when you just, spin in a calcium chloride bath, and then we're collecting here the fibers that are spun off of this spinneret. And here's just pseudo coloring showing that these microtissues are indeed aligned. And here again, showing that we can measure the alignment, and here printing in 2 different orientations. And then the other thing we've had to do, of course, is show that we had to recreate these printing trajectories using an XYZ type plotting machine. How can you recreate all of these complex geometry and the trajectories that you would need to print at, and in what sequence they would need to be printed at in order to not entangle everything? Okay, so then this leads me to one last thing, and that is I got a robotics student. Do I know anything about robotics? Absolutely zero. But I got a robotics student to design for us an automated winding device to wind cell fibers. So this is all brand new stuff, not published, but it has been disclosed. And the requirements for this is you need multiple axes of motion, tension management. You need to be able to work in the context of fluids and have moisture control. So we got this robot arm. We've got some other ones in the lab now too. And we created these test frames for winding just to make sure that we could wind in all axes without entangling the fiber. And then also in a curved structure to see, how much could you wrap around before it falls or slips off. And then here we have the construct of the gastroesophageal junction as a test frame. And here we did some simulation-based planning. First, we did it manually to figure out what the trajectories are that we need to code for the machine. And we created this tension management system here. There's a little weight. And then set it up like this. Here's the robot arm, and here I'll show you how it works. And I know that videos don't play well, probably for you, so you might have to squint and hallucinate a little bit, see exactly how it works, but hopefully you can see how it's going. And now we're trying to miniaturize this more, and but it's working smoothly, so I apologize if it's working a little jittery from your end, how you're seeing it on Teams here. So this is just showing you, as we improve the process, how much better the winding got. So the red is kind of like our final algorithms. And then here's showing actually fiber that contains pre-aligned cells inside it, muscle cells, showing you how we can do all these different wrappings around the frame. And then we've been evaluating how accurate the placement is. And the placement is really dependent on the tension with which it's winding. So we can control the speed, we can control, you know, how much it takes for each change in trajectory. And so we can wind, you know, pretty big sizes now too. And also the code, we've made it so that it's, you know, accessible cross-platform. So this is just my group here. And then I just want to put in a plug for our 3D bioprinting facility. We've got many, many different types of technologies in our lab, as you see here. And so feel free to look on our website and contact us. We do training, we participate collaboratively in projects. We do, you know, beta testing of materials as well as hardware. So yeah, thank you for your time. Sorry if I went a couple minutes over.
Thank you. That was excellent, and it's really fascinating, and thank you for sharing unpublished data as well. So now the floor is open for questions for all three speakers. Please come in. Maybe perhaps I can kind of start off the conversation, and folks can also place in the chat as well. I guess maybe for a little bit for all the speakers, but I guess a great point that Angela brought up was really related to the concept of endothelialization versus vascularization. Do you feel, you know, in terms of, are they directly proportionate to each other? Meaning, oftentimes with endothelialization, you mentioned tropism. Are there areas of denuded basement membrane even after endothelialization? Is that contributing to difficulty in vascularization, and how can we get around it? And, you know, certainly Adam and Kyle, you know, what your thoughts are on that as well.
Yeah, well, certainly it contributes to thrombogenicity, right? If the area isn't totally covered. But if you put in, you know, the right cell type that has the capacity to differentiate and fill in those spaces with time before you then, you know, challenge it with actual blood to see if it'll thrombose, then, you know, I think there's a way to do that. Of course, the other challenge is, you know, we're creating vasculature that can't respond to, you know, dilators, right, and constrictors, so you can't change pressure within it. So that's another challenge, how to incorporate all that. So, you know, for a lot of these organs, we're able to do, let's call it the simple parts, right, or even in, you know, incorporating the muscle cells. But then there are so many other cell types that are required for proper functionality of the tissue. But then again, that begs the question, okay, how much function do we really need to improve a patient's quality of life? So there's just, it's such a complex issue. Yeah, but certainly it really, I'll say, annoys me when people, you know, publish papers about, especially when it comes to in vitro modeling, and they'll say, you know, vascularized tissue, and it's not. They've basically thrown endothelial cells in the mix. You know, you can see, you know, that they'll self-assemble into small little capillary-like looking things, but then it's not perfusable. So to me, that's not vascularization. I don't know what everybody else thinks, but that's kind of like a pet peeve of mine.
Yeah, I mean, I think that's quite true, Angela. I'll also add, I think the field as a whole is, I think the jury's out, even in terms of how to potentially build, say, vasculature with bioprinting, right? I mean, I think we heard some examples where folks are trying to recreate capillary structure with biofabrication techniques all the way down to the capillary scale, right, with the most sophisticated technology possible. We're taking a slightly different approach, right? Angiogenesis happens all day long in our bodies naturally, right? So the cells are pretty good at the self-assembly at maybe up to about 100 micron diameter vessels, but not above that. So our approach has been to try to blend it, right? So don't try to fix things that cells already know how to do, but try to address, you know, the length scales at which those processes start to fail, which is the larger caliber vessels, right? But then the challenge becomes, you know, marrying up those two, which no one has really solved effectively either. So I think what'll be really exciting over the next, say, 5 probably years is really the breadth of research in that area, really trying to understand what are the most promising approaches to drive microvascularization, which, you know, right, to be honest, has been the Achilles heel of tissue engineering since its inception 35 years ago. So, you know, hard problems. I do think biofabrication and 3D printing, though, is a path towards solving that, which is why it's so exciting.
So I'm not sure if I can speak to the greater field and mechanical properties, but because we work at the interface, we'd like to see like what type of molecules we might be able to localize there to get that to work better. So a lot more work on the cell culture level, and maybe there is a formula of some type of growth factors or something. We can create a more enhanced interface that looks better and then move back to the 3D prints once we've figured that out. So that's kind of what I'm interested in doing, which is why we're sort of working on mixing and matching so many different bioconjugates at that interface. And then there's the other side of the coin, as Angela mentioned, well, what's happening with the other cells? And in the liver and kidney, it's a filter. But like most organs, there's something happening there. So, the endothelium is just one part of it. So are we actually getting filtration going through? Is it blood going through? So the thrombosis is one part of it, and which is why we're working with heparin, right? And that works quite well. But the other side of the coin is, okay, we have all these other complicated cells, and each organ has its mixtures of cells, right? So once we figure out something, that's the best case we can with the tools we have, and hopefully we can build a bigger toolbox as time goes on. There's more conjugates or peptides we can discover that'll help us with that. Then comes in, well, what happens with the liver cells? What happens with the glomerular system as opposed to these other systems? So, you can make an endothelium, but like what's happening on the other side? Each organ and each subsection of the organ's its own challenge. So you kind of have to systematically work your way up and then go back and forth or cycle back and forth. So device development might be a thing,
Yeah. And the degree of vascularization that we need too is also, dependent on the cell density that we have and the thickness of the constructs that we're building, right? Everybody's heard all that. And so the angiogenesis, like what Adam was saying, is right. Like we have to wait for angiogenesis to happen to get, the proper types of surface coverage, let's call it, or coverage within the entire construct. But then we need to think about, okay, maybe we should make things less dense so the oxygen needs aren't quite as high. And then, how do you keep things alive while the angiogenesis is happening? So, but then I, always pose this to my students and I say, yeah, well, a whole mouse can be developed in 21 days in a utero. So somehow it can happen. We just don't totally understand it yet.
One question to y'all. I think it's probably pretty easy to look at bulk flow, but how do y'all like to measure microvascular flow? Have you used lectins, other fluorescent tracers? How do you kind of assess that microvascular flow that's oftentimes very critical, getting down at the single cell level?
Yeah, I mean, we haven't, I haven't directly evaluated flow, but by evaluating barrier integrity, by looking at, for example, leakage, right? Barrier leakage, like for example, in the lungs, you can look at how much interstitial weight is increasing by, which would then tell you that, at the capillary level, you have a lot of leak. That's all we've been able to do. We haven't looked at actual flow.
Yeah, I know in our in vitro, like when we validate flow in vitro, at least, where it's obviously a lot easier. We're either doing fluorescent dyes, right, which is just both flowing in, but also the diffusion out of vasculature, right, which is obviously pretty important for nutrient transport. We do small fluorescent beads, so we can actually track flow, velocity, and vectors. We've also been trying to use like OCT-based Doppler type techniques as well. So, but and that all gets a lot trickier, obviously, if we're talking in vivo in an animal.
Yeah, it's certainly better for larger vessels. Much easier.
I think you're muted, Jason.
Yeah, sorry. Much easier for larger vessels, but insofar you haven't tried lectins before, is that right?
We've just done
beads. I think is probably the most common from microvascular assessment.
Yeah, we've done microbeads, but that's it.
We've done lectin tail vein injections, when we have an implant in vivo to look to confirm microvasculature. So yeah, we've shown in our acellular scaffolds when we implant them, we get pretty rapid, vascular ingrowth. that's due to a microporosity that we purposely introduced into our scaffolds to help drive that. So.
Yeah, this is Jay Huang. If I could chime in on that, we've used lectin tracing in the past not to quantitate flow, but to map out perfused vessel segments and areas. Well, you introduce the lectin, and then they'll label vasculature, and a lot of times, because that vascular bed is very dynamic, you'll even have regions. This isn't a normal healthy tissue that might be shut down versus not. So it's important that a lectin introduced through the circulation is only going to label those vessels that are open to that perfusion. Many times that's what you're after, but it can be problematic if you're using that approach to identify all vessel segments that might be in the tissue, because we know that other regions, and Angela had addressed this before, due to vasoactivity, et cetera, or vasospasm, depending on the treatment, might shut that down. So we tend to use a lot of different approaches, even including laser Doppler to look at blushes of perfused areas, et cetera. I've always felt that a lot of the in vivo microcirculation flow dynamic assessment methods could be applied to these built tissue constructs, whether it's in vivo or even implanted as well.
Thank you. Any other questions, speakers?
I got one last, perhaps conceptual question to whoever feels like answering, which is this idea that, say, starting from scratch to design and engineer a tissue using the adult final adapted, matured tissue as your blueprint, but recognizing that a lot of our approaches involve the evolution, for lack of a better word, of that tissue construct, et cetera. Is it better to start with a precursor design and then condition or react or drive the final outcome with the idea that once it goes in the patient, it will finish? Or does building a construct, designing and building a construct the way it is in the adult body, recognizing that's not going to get there until a while? I'm just curious to know what the thinking is on where the designing input should start from.
I mean, Jay, I think all options are on the table as of right now. I don't think we know. I mean, that's one reason we started building like essentially embryonic stage organs, because they're so much simpler in structure, complexity, and cell types, because we have to start building stuff to actually, I think, answer those questions, right? So yeah, I think that's very much the research phase we're sitting in right now. I do think that's a reason to be excited for all the trainees on the call, because there's at least a decade plus or more of that work to be done before I think we've got any definitive answers. But that's just my take.
Yeah, I think it goes hand in hand with the cells to be used.
Yeah, I've been thinking that in the process of trying to build a tissue, we're learning a tremendous amount about tissue biology. Yeah. And that will likely in the end help finish the job.
Yeah.
Questions?
All right,
we're right at the hour.
Yeah.
Right, Kimberly?
Yeah, we're right at the hour. So again, thank you very much, speakers. I learned a lot. And I recognize that organ biofabrication is definitely in the future, but plenty more work to do. And I think that's the case with everything that's happening in the field, right? As we learn, we get to do things better, and as biomaterials improve and our understanding of cell biology, then we just get better. Basically, we're kind of just learning from biology, which we should always be doing. And I want to thank Sandeep for moderating our session, and I want to thank all the speakers today for sharing their expertise. It's been quite a long day, but I think I don't think there are many meetings where you would get this kind of diversity of talks and presentations and questions, and I like to think of this space as a safe space to really get those ideas on the table and kind of share each other's challenges, because maybe someone has worked it out in their lab. So again, really, thanks everybody. So Monica has posted, I guess that's our post-meeting survey. In case you're not coming back tomorrow, so we know whether you enjoyed this, whether you would like to present your work, if you're in the audience, if you'd like to say moderate or be part of this. I'm not sure what this is, but it's definitely a community to just discuss challenges. And as I watch the symposium agenda, but also what I'm hearing is I do think there's a lot more to be done, obviously, in our biomaterial space as well. And maybe I think that speaks to Kyle and what he's doing, right, is that really sophisticated biomaterials may in fact help us kind of leverage into sort of next generation organs and things like that. So again, thank you everybody. And we do have the opportunity to have more breakouts, but I mean, that's really up to the group here, or if everybody just leaves, that's okay too. I think it's been quite a long session. I just feel sometimes that we kind of end conversations where people might be suddenly waking up and thinking of their own questions. So I'm not sure what the group wants to do. You're welcome to stay, you're welcome to go, or we can open up the breakout rooms. It depends. So
maybe just a brief introduction about tomorrow's session. I think maybe Nicanor or Kimberly want to provide maybe a teaser for tomorrow.
Nicanor, you want to go ahead?
What is going to be tomorrow?
Yeah.
I think it was summarized a little bit in Kimberly's presentation, but here is what we have. We will have the first session tomorrow morning. We'll address analytical technologies for biofabrication, insisting on other aspects which are not mainly biology, but are the technology associated, including computer simulation, and that will be Brent Craven prepared to describe the efforts in this direction with implication for product authorization from FDA. And then Alex Chortos will give an overview of application of biosensors in biofabrication. Greta from NIST will tell us about real-time quality assessments for this. Then we'll have session 2, funding of the lab to market transition. Well, as I mentioned, we'll have a talk from Army. Julie Lenzer will share with us experience in the partnership regarding government and industrial and academic partnerships in this regard. And 2 more other contributions. Then we'll have a commercialization of the biofabricated products in the context of specific context of VA with regulatory aspects and also advice from our specialized experts on intellectual property and other regulatory aspects. And this is pretty much what is going to be tomorrow. So.
Great, that's all very exciting. I think particularly kind of the regulatory aspects of things. So.
Yeah, it was sort of the homework from last time, questions that, how do government scientists leverage their discoveries sort of in a commercial sense, and how to work safely with industry and things like that. So we'll have some expertise on panel, and they'll be able to ask those more detailed questions, right? Because we do think that it takes a combination of, sort of a partnership, public-private partnership, to get to work in this complex space. All right, everyone. So Nicanor and I will open this up. Nicanor, we're going to open the breakout groups. Folks are welcome to drop. If nobody shows up in my breakout room, I'm going to be in the skin breakout. Then it's just a question of if people want to continue their conversations, because we hate to kind of cut you off if some really good ideas are percolating. So, and we'll see everybody tomorrow, noon Eastern.
So we'll close this room and open the others.
This room doesn't close until the last person leaves. So
Okay.
people can click the link in the chat if they want to join a breakout, but they're very, very welcome to go and to leave as well. And we're not going to report back, right? This is just kind of this is the tail. This is the tail of the conference, okay? So.
Well, thanks, Nora and Kimberly for organizing it, and I'll join in tomorrow between patients and clinic. Thank you. It's all very exciting.
Hopefully we'll have some recordings. I'm not sure if we successfully recorded everybody, but hopefully we'll have some recordings that we can share if you miss talks you're interested in. All right, I'm gonna sort of jump to a breakout, I think, for a few minutes. Monica, are you still there? You should be sleeping.